Hello, my name is Amanda Herried, and today my team and I will be presenting on our novel technology of polymer cartilage. To give you a quick pitch as to what our technology is trying to solve and how it can help numerous patients in the future, we created this little story. Once upon a time, there was a woman named Kathy who suffered from severe knee osteoarthritis. Every day, Kathy would suffer from debilitating pain in her knees, preventing her from walking even the shortest of distances. One day, a group of engineers came up with a novel treatment for Kathy's osteoarthritis that involved implanting synthetic polymer cartilage. Because of that, Kathy didn't need corrective surgeries every few years, and because of that, Kathy was able to live her life pain-free. Until finally, orthopedic surgeons everywhere can offer their patients a long-term alternative treatment for managing their osteoarthritis. Our proposed technology is a novel mesenchymal stem cell-seeded polyethylene glycol hydrogel matrix that will act as a long-term implant that can replace damaged cartilage. Since each implant will need to be customized for each patient, we will be using MRI imaging in order to visualize the specific size and shape of the cartilage defect we intend to replace. The advantage of using a hydrogel matrix is its low friction coefficient that allows it to mimic the lubrication characteristic of natural articular cartilage. Additionally, polyethylene glycol is cheap to mass produce, and there already exists many well-documented and currently used manufacturing methods. Finally, using a polymer as a cartilage substitute is advantageous compared to, say, a ceramic or a metal, because polymers have easily tunable properties that allow them to be synthesized with application-specific characteristics such as density, biocompatibility, and even thermal characteristics. We also propose to seed our PEG hydrogel matrix with mesenchymal stem cells, as these cells have the ability to differentiate into chondrogenic phenotypes, as can be seen in this bottom figure. These chondrogenic phenotypes can then naturally form new cartilage over time. Mesenchymal stem cell therapies have been shown experimentally to produce hyaline cartilage. That is the specific type of cartilage that is found in articulating joints. Producing hyaline cartilage is something that even other current cartilage repair technologies such as microfracture, osteochondral plugs, and autologous chondrocyte implantation all struggle to do. Additionally, by using mesenchymal stem cell therapy, we have the option of using an autograft or allograft, as mesenchymal stem cells have the ability to be harvested from multiple locations, such as bone marrow, umbilical cord blood, or adipose tissue.
As mentioned, there's currently a multitude of technologies that exist to repair damaged cartilage. Some of the most popular forms include microfracture, autologous chondrocyte implantation, or ACI, matrix-induced ACI, and osteochondral allograft transplantation. All are characterized by several notable disadvantages, such as transplant rejection and the formation of fibrous cartilage instead of the hyaline cartilage. As discussed before, our proposed solution of polymer cartilage consists of mesenchymal stem cell seeded PEG hydrogel implant. This brings many advantageous qualities to the table, such as versatility by being capable of many configurations and geometries and being very cost effective. Polymers and hydrogels are commonly used today and manufactured for biomedical applications. The clinical impact from this technology could rapidly reshape the cartilage repair industry by delivering a low cost yet highly flexible technology to a wide range of customers. Ideally, our solution will offer better long-term success rates than other available technologies and decrease the need for secondary repair surgeries. Next slide, please. The market for our technology shows considerable opportunity. Compound annual growth rate of regenerative therapies that involve cells, tissue engineering, and cartilage is expected to be 12.5%, from 255 million in 2015 to 460 million by 2020. Currently, the market is still in its growth stage with good opportunities for new technologies like ours to enter the market and secure a significant portion of the market share. Other currently existing technologies and competitors do exist, however. To note, some of the similar patented technologies are Opticure, Inductos, Cordos, Helos bone graft substitute, and Immix extenders. The price of these products generally range from about $2,500 to $5,000. The two major causes of the cartilage injury are osteoarthritis and traumatic injury. According to the Arthritis Foundation, degenerative joint diseases such as osteoarthritis is estimated to affect more than 130 million people worldwide by 2050. In addition, market drivers include new sources of stem cells and standardization of stem cell therapies in clinical practice and increase in the prevalence of conditions treated. There exist many barriers to enter the market, however. Some of these include a lack of research related to clinical applications and long-term patient outcomes, leading to an inability to translate research into usable technologies in a clinical setting. Finally, the exuberant costs related to getting technology FDA approved and the related cost of the regulatory pathway pose a large problem to the development of new therapeutics. Slide, please.
We found a number of patents in our search that could be closer or in some way conflicting with our patent. But today we are presenting the ones that had the highest rates in terms of ones that were more closer to our patent claims. One of them is the amphiolic peptide or peptide material. This is useful for in vitro or in-site biomineralization and induces bone repair. This present innovation presents a peptide which uses a gel or certain other gel in accordance to the innovation, comprises of cells within the adhesion of a peptide matrix. It conflicts with our patent in use of a gel. So the second selected patent or in our search was the tough hydrogel. That comprises of a hydrogel. This innovation provides a method for implantation as well as a tough hydrogel in addition to administration of a gel. The method involves cross-linking, hydrosolidification, and pre-gel. This could infringe with our claims since we could use the same mass manufacturization processes. Next slide, please.
Polymer cartilage is a class 3 device because it's an implant that's intended to be left in the patient's body for a long time, ideally for more than 10 years. It is also used to cure or mitigate the impairment of human health. And for 21 CFR 812, we need to have an ID approved by the FDA for high-risk devices, including ours, before doing any clinical trials. If our device is successful at the clinical trials and ready for the market, we need to complete a pre-market application. We can't use the 510K pre-market notification pathway as we can't claim substantial equivalence due to mesenchymal stem cells raising different safety concerns such as origin, production, and storage, even though our device has the same intended use as the device is already on the market. Next slide, please. We first want to learn more about the size of the lesions in the injured area so that we can have the right amount of polymer, and MRI is a really good non-invasive method for doing that. For the MRI scans, we would like to get the images with contrast, as contrast makes the structures in the joint easily distinguishable. First number on top is for the joints of upper extremities, such as elbows and shoulders, and the second number is for the joints of lower extremities, such as hips and knees. Reimbursement for MRI scans with contrast is around $366. For the surgical procedure for placing the implant, the closest HCPCS level 1 or CPT we could find was 27,412, which is used for autologous chondrocyte implants. Reimbursement for this procedure is around $1,175. We do not have a HCPCS level 2 code for this device yet, as the use of mesenchymal stem cells is still on its experimental or investigational stages. Next slide, please. My name is Ozge Buyukcelik, and I have an undergraduate degree in biomedical engineering, and I'm doing my master's on vagus nerve stimulation for treatment of epilepsy.
My name is Amanda Herried, and I have an undergraduate degree in biomedical engineering, along with a minor in polymer science, and I'm currently pursuing a master's degree also in biomedical engineering.
My name is Zoe Sekyonda. I have an undergraduate degree in biomedical engineering. I'm currently pursuing a PhD in biomedical engineering with a focus on translational research.
And I'm Daniel Stasik. I have my undergraduate degree in biology and I'm pursuing a master's in biomedical engineering.
Thank you so much for listening to our presentation today.